Good evening, dear chairman, speakers, moderators, and attendees. Hope you have spent a great day with fruitful new knowledge. Here is Reem, Pediatric Oncology Clinical Pharmacist at Burj Al Arab Main University Hospital, with my session today, Drug Modifications in the Pharmacogenomics Era. Let's start. What does pharmacogenomics mean? It is a broad term which studies how all of the genes or multiple genes can influence the response to drugs. Humans are 99.9% genetically identical, and only the 0.1%, the remaining, is referring to the 3 million differences. Those differences are called polymorphisms, but before going through the polymorphisms, we should know the language of pharmacogenomics. The nucleotides, which are A and G and C and T, are the alphabet of the DNA. Each codon is made up of 3 nucleotides, and the gene is made up of a group of codons, and the chromosome is the whole body carrying the genes. And the chromosome is carrying the genetic material and our final DNA, which is the genome. If you want to go through testing for polymorphisms or genetic testing, so we are having common laboratory methods for pharmacogenomics testing. Number 1, PCR. Number 2, Pyro Sequencing, and the Tacman and the DHPLC. Let's start with the PCR. We will take blood cell or blood sample, and to take blood cells from this blood sample or a cell from the buccal cavity, which are commonly collected to obtain a DNA sample. Once DNA is collected, the genomic DNA must be isolated from other cellular material, and then specific region of interest must be identified and amplified and performed via polymerase chain reaction. And then the verification process is to do gel electrophoresis, which is often performed after PCR to verify that PCR was successful and that the amplified target sequence is the correct size. PCR coupled with restriction fragment length polymorphism analysis, which is a conventional and not automated genotyping method, which is very practical for laboratories that genotype a limited number of samples. On the other hand, the Pyro Sequencing, which is an automated method, it is to take the desired DNA and produce pyrophosphate, which undergoes some enzymatic reactions releasing ATP. ATP undergoes metabolism by luciferase enzyme and causing light emission. And Tacman process. Tacman is another technique used to find SNPs and indels. What are SNPs and what are indels? SNPs are single nucleotide polymorphisms, and indels are insertion and deletions polymorphisms. This process is a quantitative PCR or real-time PCR. Using this technique, you can follow the concentration of DNA increase after each PCR cycle based on the fluorescence of the probe, and each probe is very specific to a very specific SNP. The last type we have DHPLC, or denaturing high performance liquid chromatography. This method is both qualitative and quantitative. It is a method of chromatography for the detection of base substitutions or deletions or insertions by using PCR amplification, denaturation, and re-annealing by heating and slow cooling, resulting in 2 different alleles. This is the final result we can find on the first line. This is the wild type, while on the mutant line we can find insertion substitution here in the gene. So we are done with the testing, and now we are having the patients or the people gene map with the gene specific mutations. So why does drug response vary? It is due to genetic variations. Number one, single nucleotide polymorphisms, or insertion or deletion of one or more nucleotide, or due to the CYP enzymes, which are the metabolizing liver enzymes mutations. So, number one, the single nucleotide polymorphisms, and we call it a SNP, single base mutation, which substitutes one nucleotide with the other. Those are the most occurring genetic differences. Between any two people, there is a SNP for every 1250 bases, but most of them have no phenotypic effect. Some of these are alleles of gene and show us the phenotype. Number two, due to insertion or deletion of one or more nucleotide. If insertion of one or more nucleotide is repeated, so it is called tandem repeats polymorphisms. And variable number of tandem repeats gives variable length of sequence motifs that are repeated in tandem in a variable copy of number. And the types is based on the size of the tandem repeats, whether it is a microsatellite or it is a mini-satellites. Microsatellites, it is short tandem repeats, dinucleotide repeats from 1 to 6 units repeated, while the mini-satellites repeat of units 14 to 100. In microsatellite, it is a dinucleotide repeat, while in mini-satellites, it shouldn't be or it must not be dinucleotide. Here is an example of insertion and deletion polymorphisms. Number one is the wild type alanine, arginine, alanine, and it is a fully functional protein. Whether the missense that arginine is substituted with lysine, and this produces a missense substitution with a less functional protein. On the other hand, the nonsense is that lysine alanine is substituted with a stop codon, so this allele is not functioning at all. Number three, the CYP enzymes, they play a major role in the drug metabolism, primarily in the liver, and can be distributed in the intestine, lung, kidney, and brain. For each CYP gene, star 1 is the wild allele. SNPs are then sequentially numbered star 2, star 3, star 17, star 28, etc. What do UM, RM, and MIM-PM stand for? UM, ultra rapid metabolizer, RM, rapid metabolizer, NM, normal metabolizer, and IM, intermediate metabolizer, while PM is the poor metabolizer. So hypocrites have a very nice quote, which is, it's far more important to know what person the disease has than what disease the person has. So, does one dose fit all the patients? On the left box, there are a group of people who are taking the same dose 100 milligram, while on the right box, after the pharmacogenomics era, the ultra-rapid metabolizer took his suitable dose 500 milligram, and the extensive and intermediate metabolizers took their suitable dose, which is 100 milligram, and the poor metabolizer took his suitable dose, which is 10 milligram for less toxicities. Pharmacogenomics and pharmacokinetics relationships. Pharmacogenomics studies the set of variations in genes related to pharmacokinetics and pharmacodynamics and their relationship with pharmacological response. In other words, pharmacogenomics studies the relationship between genetic information and the risk of incidence of a determined event, or the genetic contribution to its incidence, understanding an event either as an adverse effect or therapeutic success. Pharmacogenetics and pharmacogenomics are tools that allow explaining and predicting variability in ADME process and association with the clinical success or therapeutic failure due to lack of effectivity or security. Both disciplines are important tools for personalized medicine. Here is a diagram to show the effect of pharmacogenomics on the ADME process. A stands for the ultra rapid metabolizer who takes the subtherapeutic dose due to the same dosing. B is our normal metabolizer or intermediate metabolizer, so he is not affected. C is the poor metabolizer who took the same dose as A and B and suffered from toxicities. So let's go through hematological malignancies and see some examples of drugs. Asparaginase-induced pancreatitis is due to the triple repeat versus double repeat. The triple repeat is haplotype star2 of asparaginase synthetase gene, resulting in remarkably higher risk of toxicities associated with the 3R or the triple repetition genotype. In contrast, the double repetition, which is the haplotype star1, had a protective effect against these adverse events. Management: those with 3R or the triple repetition haplotype star2 asparaginase synthetase predicting high risk of pancreatitis. So, number one in the management, to close monitoring for any signs of pancreatitis. What are the signs of pancreatitis we can see in the patient? Typical abdominal pain associated with pancreatitis, persistent and radiating back pain. Level of serum amylase or lipase is 3 times higher than the upper normal level. And typical signs of pancreatitis on imaging, either ultrasound or computed tomography, the CT scans. And the drug management, that we can give octreotide as supportive care for asparaginase pancreatitis plus 50% dose reduction of the asparaginase dose. Number 2, in the hematological malignancies also we can find gene variants in the TPMT enzyme, which is the thiopurine methyl transferase. STAR2, STAR3A, STAR3B, those are alleles of losing functions. The percentage of this gene variation is about 5.8 to 15.5% in the individuals who are carrying an actionable TPMT. IM stands for intermediate metabolizer. He is an individual who is carrying one normal function allele and one no function allele. The example is STAR1, STAR2. STAR1 is the example of functional allele and STAR2 is the example for non-functional allele. So one with complete sense and one with missense, so both make the intermediate metabolizer, or STAR1, STAR-A, STAR1, STAR3B, STAR1, STAR3C, or STAR1, STAR4. The management here is to start with 30 to 80% of the normal dose. Example, we are giving mercaptopurine on dose of 75 mg per meter square to start with 25 to 60 mg per meter square. And for dose adjustment, we should wait for 2 to 4 weeks for the drug to reach its steady state. Alright. On the other hand, we have the poor metabolizer, who is an individual who is carrying 2 no function alleles. Example, STAR3A, STAR3A, STAR2, STAR3A, STAR3A, STAR3C, or STAR3C, STAR4, STAR2, STAR3C, or STAR3A, STAR4. And the management here is to start with drastically reduced doses. Reduce the daily dose by 10 folds and reduce frequency to thrice weekly instead of daily. Reducing by 10 folds means that to give about 10 mg per meter square per day, given only 3 days per week. And also for any dose adjustment, here we should wait for 4 to 6 weeks to reach the steady state, and then to decide whether we are going through dose adjustment or not, because this patient is poor metabolizer. But some patients may suffer from mercaptopurine toxicity although normal TPMT function, and this is due to another genetic variation in the NOD-T15 gene, which is Nodix hydrolase enzyme 15. Here the intermediate metabolizer is STAR1, STAR2, or STAR1, STAR3, And the management here is to give also about 30 to 80% of the dose. So if the dose is 75 milligram per meter square, to start with 25 to 60 milligram per meter square, and for any dose adjustment we should wait for 2 to 4 weeks. On the other hand, here the poor metabolizer is star 2, star 5 or star 3, star 6, but the management here is like the intermediate metabolizer, to start 30 or 80% also of the dose. So if the dose is 75 milligram per meter square, to start with 25 to 60 and to allow 2 to 4 weeks to reach the steady state. This in conclusion reveals that this enzyme activity on mercaptopurine thioguanine is not as potent as the TPMT effect. Last but not least, in the hematological malignancies, methotrexate, the gene in charge here is the methylene tetrahydrofolate reductase, or MTHFR. The mutation in charge here is C677T. What does this mean? This means that C is the wild type allele, while T is the mutant type allele. And in the mutation, T replaces C at the 677 site. So instead of being CC allele, it is TT allele. Or sometimes it could be CT allele, which is also would be intermediate metabolizer. The percentage of this mutation is about 10 to 12% in the Caucasians. CC genotype has the best prognosis, less myelosuppression, and less nephrotoxicity and hepatotoxicity in transaminases number. The management here is to start with methotrexate dose reduction by 25%, aggressive alkalinization by doubling the amount of sodium bicarbonate, and increasing the rate of infusion to decrease the toxicity. Let's go through the solid tumors. AP protocol in osteosarcoma. AP stands for anthracycline platinum. Cisplatin is considered one of the platinum family. Gene here is the TPMT gene, thiopurine methyl transferase. The mutation here is, or the mutant alleles here are star 2, star 3A, star 3B, and star 3C. TPMT star 2 is functionally inactive in the pediatric population. Therefore, reduced enzyme activity and more drug in the blood. The expected side effect here is ototoxicity. 92% of patients carrying this variant develop hearing loss. And the management here is to add otoprotectant drugs like amifostin or sodium thiosulfate. Number 2, the irinotecan. Second line setting in neuroblastoma and Ewing sarcoma, irinotecan timodal. Irinotecan can have very severe neutropenia due to the gene mutation of the UGT1A1, which is the UDP glucouranosyl transferase, one of the liver glucuronidation enzymes. The poor metabolizer mutation is star 28, star 28. The percentage in the Caucasians is about 29 to 45%. Serious life-threatening adverse events occur more often in patients with this genetic variation. The genetic variation reduces the conversion of irinotecan to its inactive metabolite, though the active irinotecan will be remaining in the blood for longer time, giving more and more toxicity and more immunosuppression, leading to severe neutropenia. The management here is to start with the 70% of the standard dose. If the patient tolerates this initial dose, the dose can be increased but guided by the neutrophil count. Let's go through the diagram of this recommendation. This is an example of diagram found on PharmGKB website. If the dose of irinotecan is less than 180 mg per meter square, so the UGT genotyping is not recommended. Also, pharmacogenomics is related to pharmacoeconomics, so we should know when to use or when to make the genotyping testing and when not. So if the dose of irinotecan is less than 180, it's not recommended at all to genotype the UGT1A1, while if the dose is 180 or more than 240, so UGT1A1 is mandatory. Whether it will give homozygous wild type *1*1 or heterozygous *1*28, which is intermediate metabolizer, or homozygous mutated *28*28, which is the poor metabolizer. Okay, and the decision here is to give dose reduction of 30% of the dose at first cycle, and then if tolerated to give or to raise the dose, but guided by the neutrophil count. The fluorouracil, which is used mainly in the colorectal cancers, is guided or is metabolized by an enzyme called the dihydropyrimidine dehydrogenase, the DPYD, the rate-limiting enzyme for 5-fluorouracil catabolism, and it is found on the short arm of chromosome 1. The types of mutations here are types *2A and *13. The toxicities expected are hematological or gastrointestinal, but not hand and foot syndrome, which is related to this mutation. Fluorouracil's prodrugs are capecitabins and tegafur. Normal metabolizer has what is so-called of AS, or the activity score. So the normal metabolizer has his activity score equals 2. Intermediate metabolizer has his activity score of 1 to 1.5, while the poor metabolizer has his activity score to be equal 0. The management in case of intermediate metabolizer activity score 1 to 1.5 is to give 50% dose reduction, while the poor metabolizer who has activity score 0, it is recommended to omit the drug totally. Some guidelines recommend to give the capecitabin or the tegafur, as they are the prodrugs, and being prodrugs will never have toxicities as the metabolite, but it is not a strong recommendation. Let's go through the supportive care. We all know that phenytoin has many drug interactions, and it is very toxic as well. CYP2C9 is the enzyme of charge for phenytoin metabolism. Poor metabolizer in phenytoin has the mutated alleles *2*2 or *2*3. The action to be done here is the same loading dose is to be administered, while we will give 50% dose reduction in the subsequent maintenance doses, with monitoring also the serum levels of phenytoin. Let's go through the antifungal like voriconazole. Voriconazole is one of the most toxic antifungals used in the cancer therapies, and it should undergo TDM periodically, mainly undergo metabolism by the CYP2C19 liver enzyme. The ultra-rapid metabolizer or the rapid metabolizer carries *17 or *1*17 alleles, and the management here is according to which guidelines you are following, whether you are following the CPEC or you are following the DPWG. If you are following the CPEC, so the CPEC recommends an alternative agent which is not dependent on the CYP2C19, like liposomal amphotericin B, while the DPWG recommends to elevate the dose by 150% of the main dose. On the other hand, the intermediate or the normal metabolizer, who is *1, *2, *1, *3, or *2, *17, is to initiate with the initial dose with the usual TDM but more frequent. And the poor metabolizer, who is *2, *3, the management here is also according to which guidelines you are following. If you are following CPEC, so it is much preferred to use the liposomal amphotericin B, or if you are following the DPWG, so 50% dose reduction will be the choice. And at the end, all patients or all cancer patients may suffer from pain, so we should know how to manage this pain for these patients. The gene in charge for tramadol or codeine is CYP2D6. The ultra rapid metabolizer is *1*1. And the management is to avoid the codeine use because of potential for serious toxicities. If opioid is warranted, so consider any other drug, not CYP2D6 candidate. On the other hand, the intermediate metabolizer, who is *4, *10, *10, *10, *41, *41, the management here is to use the codeine label recommended age or weight specific dosing, or another agent which is not a candidate of CYP2D6. While poor metabolizer *4, *4, or *5, *5, or *5, *6, the management here is to avoid codeine use at all because it will have a possibility of diminished analgesia. Those are the references, and mainly we are working from Pharmacogenomics and from CPEC. Thank you so much for all, and I hope I have reached your aspirations and hope it was a very fruitful lecture. Have a nice day with more and more amazing lectures.